Hello, everyone. I'm Dr. Matthew Williams. I'm one of the instructors for Biology 111 lecture and lab. So, again, this semester you're going to be seeing several instructors giving these intros, which is nice. You get to see not just your normal instructor, but some of us other instructors also. So, in week 2, lab 2, we're going to be looking at biological molecules. So, the biological molecules, you'll be either already learning about them in lecture or you'll be getting there very soon. So that's one of the main things we want you all to get from Biology 111 is understanding these biological molecules. So what I mean by biological molecules, typically when we refer to biological molecules, we're talking about the organic molecules. So there are lots of molecules of biology, but when we refer to biological molecules, it's mostly talking about 4 categories. And the 4 categories we're talking about are all organic. Again, the phrase organic means those that have consist primarily of carbon and attached hydrogens to those carbons. Again, the 4 main ones we discuss are carbohydrates, lipids, proteins, and nucleic acids. In the second lab, in lab 2, we're really going to look at 3 of those, 3 out of 4 of those biological molecules. We're going to look at carbohydrates, lipids, and proteins. Nucleic acids are very, very important. So things like DNA and RNA. Again, DNA is what really makes up the set of instructions that says who you are and who that oak tree is outside and all of that. So they're really, really important. But we're just in this lab, we're looking at 3 out of 4 of these. In addition to those 3 biological molecules, we do look at some inorganic molecules that are important in biology also. So we look at vitamins. In this lab, we look at vitamin C, and we also look at water. I mean, water is a molecule that is very, very important to biology. So we kind of use the water as a control, but again, realize it is an important molecule for biology also. So when you're looking through your handout, you'll notice we talk on the carbohydrates and lipids and proteins, and make sure you read that information. It's really good information. It helps sure up what you're learning in lecture, hopefully. Then we go into talk, again, those are going to be those 3 are the, going to make up the 3 main, 3 of the 4 main biological molecules. And then we also discuss in the handout water, as I mentioned, is very important. And then vitamins and minerals also. So the lab it's broken up into 3 parts. And I'll have 3 other videos that go with this one. I actually created those first because I knew I would mess up on some things. So I'm going to try to point out some of the things that I misspoke on or messed up on here in this intro. And so that when you get to those videos, I can hopefully clarify that now so you can be watching for my mistakes in those videos and then get them sorted out. So rather than redoing a whole 13, 14, 20-minute video, however long they were, I'll point them out here. So again, 3 main parts. You're going to first do a qualitative analysis of biological molecules. You're going to look at 3 of those. You're going to look at the carbohydrates, lipids, and proteins. And then again, you're going to look at also the vitamins and water. So the qualitative analysis, you're going to have those molecules, and then you're also going to have some indicator solutions where you try to see which indicator solution works best for those molecules. You know, if you wanted to identify protein, would you use Sudan, or would you use iodine, or would you use the copper sulfate reagents? And sometimes it's very useful to be able to identify what's in the solution. So if you've got something like, mystery solution from patient A, is this, full of protein, and how much protein? Is it full of lipids? How much lipid? Again, if you've got protein in the wrong place, or you got a really high level of protein in the wrong place at the wrong time, that can be indicative of, something's wrong. So, so many examples of what we could use that for. Again, sometimes, you're trying to make protein. So say you're working in industrial microbiology and you want to make some protein, or you want to make some lipid, and you want to know, am I getting that, or how much is there? You might use an indicator solution to say, hey, yes, you are getting this lipid, or you are getting this vitamin. and then you could do a quantitative analysis to see how much. Again, today with those indicator solutions, or in this lab, you're going to just get a qualitative insight to that for part 1. That's mistake number 1 I made. I was calling that part 2. So the qualitative analysis is part 1. Part 2, and the real part 2, is a quantitative analysis. Again, here we're going to use a separate setup, all new test tubes, entirely separate of part 1, to do a quantitative analysis. Now here you're not going to be using biological molecules. What you're actually going to use is red food dye, because it makes it really easy for us to see if you do things right or wrong in part 3. But the real part 2 is that quantitative analysis where you're going to set up serial dilutions. So serial dilutions is anytime you prepare a dilution, you have a concentration. So your concentration is going to be a ratio of the amount of some substance dissolved into some solution. So the examples we give in the handout is something like 2 grams of sodium chloride, so table salt. So 2 grams of sodium chloride dissolved into 100 milliliters of water, or 3 drops of food dye in 8 milliliters of water. That's the concentration. So some students have a hard time realizing sometimes it's that simple. If we're asking you, how many drops of food dye per mils of water, it's just what you did. So, if you're adding 1 drop of food dye per 8 mils of water, that's the concentration. 1 drop food dye, 8 milliliters of water. If you were to add double the amount of water there was there, you'd actually be halving the concentration. So if you had 8 milliliters of water and now you add 8 more milliliters of water, that 1 drop of red food dye is less concentrated. So because there's more water, but there's less food dye per that ratio. So realize that some of the questions we ask you in this lab refer to that. Again, you do have a lab exam that comes up eventually, and some of those questions are on that also, refer to concentration. So if you have questions about concentration and you're struggling with that, make sure you check with your instructor, find me, talk to me about it, and make sure you understand that, how concentration works. But what you're going to be doing is diluting things. So if you add that water, so I mentioned that example of 1 drop of red food dye into 8 milliliters of water. If you want to dilute that, you could add more water. So let's say we add another 8 milliliters of water. Now it's that 1 drop in 16 milliliters of water. It's more dilute. So sometimes you want to do that to figure out, well, in our case, we're going to figure out how good you are at diluting things, how good you are at working with the graduated pipette. And the way we're going to do that is we're going to have you do those dilutions. You're going to do 2-fold dilutions. So you're going to take some concentration of red food dye to water. You're going to add more water to it in the second test tube, and then you're going to add more water to it by transferring it to a third test tube. That's where I think only my second mistake comes in with the videos. With this setup, as you're reading through your handout, you'll notice, and watching the other video, you'll notice that it talks about 4 test tubes. In that video, I tell you to dilute it all the way out to the 4th test tube. Don't touch the 4th test tube. Don't add food dye to the 4th test tube. Read your handout and understand that. So in that one, I say, okay, take your stock of food dye. You'll have a nice red test tube full of red food dye, and I tell you to put it into the second one and put it in the third one, and I tell you mistakenly to put it in the 4th one. Don't do that. You're only going to put it all the way down to test tube 3, and your handout tells you the right way to do it. So tube 4, you don't add anything to other than the water, so pay attention to that. And then you'll take those test tubes that you've done the serial dilution with, and you'll use them in part 3. So part 3 is where we use a instrument to measure the amount of red that's in your test tubes that you set up in part 2. So again, if you start off with just straight food dye, it's going to be really, really bright red. If you dilute it with water, it's going to get to some shades of pink. And the more you dilute it, the more pink it gets. And if we diluted it all the way out, it ended up being clear. But what is red? So what is the color red? Why is something red? It's because it's transmitting the color red back to you. So if you have a test tube that is full of red food dye. The reason it's red and you're seeing red is it's actually absorbing all of the light waves except red, and it's transmitting red back to you. So if you want to know how red something is, you can measure the transmittance of how much red is coming back, or you could measure the absorbance of how much of other colors are being absorbed. So that's what we're actually going to do. We're going to use a spectrophotometer to measure the absorbance. So take a look at the table that's in your handout for part 3. And you'll notice it has wavelengths in it. It's also going to have a color, and the color something appears to be. So again, those wavelengths and the color refer to the light that's being absorbed by the solution. So if you have something that's red, the colors being absorbed by that are actually the opposite of red. So if you look at that little color wheel on the right in your handout, you'll notice that the main color directly across from red, the opposite color to red, is green. So if we want to know how red something is, we're going to measure the absorbance of green. Because the red's being transmitted back, and I said that we want to measure how much is being absorbed to figure out how much red it is. We could do either one, but we have a spec that's going to allow us to read the absorbance, so we're going to measure how much green is being absorbed. So we're going to set the spectrophotometer to measure how much green light is being absorbed. And by telling us how much green light is being absorbed, it's going to tell us how much red is reflected, hence how red it is. So if it's really, really bright red, you would get a really high number for the absorbance of green light. If it's slightly pink, you're going to absorb less green light. So pay attention to that. Understand why that is, that we're measuring absorbance versus transmittance, is because that's what our spectrophotometer is reading. It's reading absorbance of light. But that's a very common measure to figure out how colorful something is to measure the absorbance of what's actually being taken into that. And then it can tell you how much is being transmitted back. So you're going to work with your instructor to set up the spectrophotometer and make sure it's set up to the right wavelengths. They're going to talk about blanking the spectrophotometer and or have you do it. Some instructors may have you do it, or again, if you have questions about that, again, you have to put your test tubes into it and blank it out. And then after you get your blank reading, you're going to measure each of the 3 test tubes that you set up in the second part of the lab. And your blank is that 4th test tube that should just have water in it. Don't listen to my other video where I say put red food dye in tube 4, keep just water in that. That ends up being your blank for this last part. You're going to get your 3 readings of your serial dilutions. We're going to see how good your dilutions were, how good you were with pipetting, and then also we're going to ask you some questions about that. Why is it, why are the numbers what the numbers are, and why would you expect that to be? So I think that covers everything for Lab 2 intro. Again, there's 3 other videos that you'll want to watch before the lab where I actually go through and show you most of how to do the lab. So again, hopefully keeping things very efficient with the lab. Again, if you have any questions, ask your instructor, find me, ask me. We're all here to help. So whatever you need, definitely use us as a resource. Quiz should be posted also, so make sure you do your pre-lab quiz. And then fortunately there's no lab report for this one. It's just the handout. Working through that handout, and then there's going to be an answer sheet you're going to fill out. So with each of the sections, there's a few questions on that answer sheet. Make sure you answer those questions on the answer sheet and get that to your instructor.